There are two types of cells, and we call them prokaryotic and eukaryotic. There are some definite differences between the cells structurally, and we do find them composing different organisms. So prokaryotic cells tend to be the simpler cells, and these are found only in organisms from the domains archaea and bacteria. They're really tiny cells, and we've talked about the features of those. When we're talking about eukaryotic cells, these are going to be larger cells, they're going to be more complex, and we tend to find them in organisms that are multicellular. It's not 100% of the time the case, but most of the time. So this is going to include kingdoms Protista, fungi, Animalia, and then also Plantae. So let's talk some about the features of the eukaryotic cells. Okay, so first off, again, if we just look at them size-wise, the prokaryotic cell is going to be extremely tiny compared to the eukaryotic cell. The eukaryotic cell is going to be bigger, and it's going to have a lot more components and organelles that we do find inside of it. If we talk about the external structures of eukaryotic cells, external structures would be things found outside of the cytoplasmic membrane. If we're talking about animal cells and protozoan cells, these are going to have a glycocalyx in many cases surrounding the cytoplasmic membrane. This is a sticky structure that's found on the outside. This sticky structure would certainly provide some protection for those cells because it is an extra layer on the outside of them. And then when we talk about fungi, algae, and plants, or other ones that are eukaryotic organisms, these are going to have cell walls. The cell wall, you can see in the picture here, it can be a really thick structure. The one that we're seeing in this image is formed between two separate cells. But this rigid structure, it is similar to what we find in bacterial cells, but it is made up of different materials. Exactly what materials it's made of depends on what cell you're talking about. If it's fungi, it's typically made of chitin. If it's a plant cell, it's typically made of cellulose. And then the algae, theirs usually are made of some cellulose as well, although they can structurally be a little bit different than what we find around the plant cells. If we talk about the functions of that cell wall, it's pretty similar to what we had with bacterial cells. It is a rigid structure found around the outside of the cell. So because it's so rigid, it is going to provide some protection. That protection can be against the environment. It can be protection against osmotic pressure, so osmotic forces, water flowing in and water flowing out, so the shrinking and the expanding. And then it also is going to help the cell to actually maintain its particular shape. If we look at the actual plasma membrane or cytoplasmic membrane of the cell, this is the general structure to it. It is really similar to what we've already looked at. It is made up of phospholipids. It will be a phospholipid bilayer, meaning two groups of them. And there's going to be this portion on the inside here that does not like water. And then where you see all the little circles on the top, and then on the bottom is where we can have water touching it. One thing that is unique about eukaryotic cells is that we can have steroids inside of the membrane. Okay, so that would be cholesterol is one example. And you can see the structure here is that you have a lot of little hexagons stuck together. There's actually a pentagon there on the bottom. So those can be found inside the membrane, and they will affect the overall what we call fluidity or the flexibility of the membrane. And then we can see it up here on the top, we can have some sugars. Those sugars, they would be on the outside side of the cell or the external cell side. And these are a lot of times important for communication between cells or recognition even between cells so that this cell can recognize this other one over here. Another thing that's unique about the membranes in eukaryotic cells is that we can get these structures called lipid rafts that form. And the lipid rafts are going to be where we get a lot of tight packing of some lipid molecules within the membrane, and it will look distinctly different and it will move distinctly separate from the rest of the membrane. These lipid rafts, they are important for just some different cellular responses when the cell is trying to respond to maybe some type of pathogen and just have this coordinated effect. We'll see that the lipid rafts will form and they may move around in some specific way inside the membrane. If we talk a little bit about how a eukaryotic cell is going to bring substances into the cell, this is accomplished using endocytosis. Endocytosis is just the bringing in of substances, and then we can categorize this a couple different ways. And phagocytosis and pinocytosis are two types of endocytosis. This picture right here shows the endocytosis. Specifically, this one would be phagocytosis. And what we see is that the cell, you can see it has kind of a different shape to it, or an irregular shape to it. And what's going to happen is that we get these kind of structures or arms that kind of form because of shape changes. And those structures are called pseudopods. And they will wrap themselves around something and engulf it. So we're really talking about an engulfing. And when it's phagocytosis, it's because the cell wants to eat that particular substance. So it's a cell's way of kind of opening up its mouth and bringing that substance in. So we can see this small little cell right here, it's getting wrapped up. We can see it partially, and then by the time we get over here, it is completely inside. And at that point, once it's inside, the cell is going to take it apart because, again, it's going to break it down for food purposes. So phagocytosis, kind of bringing it in for eating. When we're talking about pinocytosis, it's a little more specific selection of a particular substance and bringing it in. But the idea is that we're still going to have this bringing in to the inside of the cell. And there will be some shape changes in the overall structure of the cell when we are completing that process, or any of these processes here. All right, if we talk about the cytoplasm of eukaryotes. So by cytoplasm, we're talking about the cytoplasmic membrane, everything that's inside of that. Eukaryotic cells can have flagella too. The difference with their flagella is that they're actually found within the cytoplasmic membrane. So we do think of them as a projection from the cell, but it's a projection that is coated with the plasma membrane. So that plasma membrane shown in blue here is actually going to cover the entire flagella. So it's really going to be stretched out beyond the outside of the cell. And this flagella is made up of a different material than what we have in bacterial cells and archaea cells. This particular one is made of a protein called tubulin. And that tubulin is made into bigger fibers, which we call microtubules. And then those microtubules get placed into a very specific characteristic arrangement for eukaryotes. And we call it a 9+2 arrangement. Well, what the 9+2 arrangement is referring to is the fact that we're going to have 2 microtubules in the middle, and then there's going to be 9 of them that are arranged around the outside. So that's the 9+2. Bacterial cells, it is going to be completely different protein, and it is also not going to have these central ones that everything else is surrounding on the outside. In this case as well, these particular flagella, they're not going to be rotating like a propeller, they're going to be waving. And that waving is what we call undulating. So there's an undulating movement which is going to help to move the entire cell. Other structures that they have are called cilia. Cilia are going to be like little hairs. So these also are internal, meaning they would be covered by cytoplasmic membrane. And if you just look in the picture here, you can see that there can be a lot of these on the outside of the cell. In general, they're shorter, and there will be many more of them. They do kind of wave around so that they do help the cell to move from one place to another. In some cases, they wave so that they kind of draw something towards the cell. If there's a particular particle that they would like to engulf by phagocytosis, they can kind of gather it by just waving and drawing it towards themselves. These are also going to be made of tubulin, just like the flagella, and it's also going to be a 9+2 arrangement. These, as we said, they beat rhythmically, the beating of them rhythmically. So it is kind of a waving, but they're just not as long, so it doesn't look as much like a waving. It is movement for the entire cell, or it is drawing a substance towards themselves. We talk about some other non-membranous organelles. So by non-membranous organelles, these do not have a membrane that's going to cover the entire surface. Ribosomes, we would have ribosomes definitely inside eukaryotes. Ribosomes are going to have the same function that they had before, and that big function was that these are going to make proteins. So they're making proteins. These particular ribosomes are going to be bigger than what we had in prokaryotic cells, and they're either floating around in the cytoplasm or in some cases, they're going to be stuck to some of the internal membranes. Specifically, we'll have them stuck on the endoplasmic reticulum, which we'll see a little bit later. The cytoskeleton would also be something found inside of the eukaryotic cytoplasm. This is going to be that fiber that extends throughout the cytoplasm, kind of makes this network inside internal skeleton, internal framework. This internal framework is going to allow us to anchor different cellular components in different places so that everything's not just randomly moving around inside the cytoplasm. It's going to function in cytoplasmic streaming, basically moving of a substance from here to there inside of the cell. That's what that means. Okay, so moving of organelles, substances inside the cell, anchoring them to particular locations, and it's also providing some structural support on the inside of the cell. Other things that we have centrioles and centrosomes. Centrioles are going to be lacking in plants, algae, and most fungi, but this would definitely be something that we would have inside of animal cells and then also in some protist cells or protozoan cells. Centrosomes contain the centrioles, and these are going to have roles in cell division, and specifically it's going to be mitosis, specific type of cell division or cell replication. And then they also have a role in forming the flagella and then also the cilia. If we talk now about the membranous organelles, all of the membranous organelles are going to be surrounded by a phospholipid bilayer, so actually phospholipids surrounding them, and we would not have any of these when we're talking about prokaryotes. So these are things that you would not find in bacterial cells, and you would not find them inside of your archaea cells. The most popular of all of these is the nucleus, and that's what you're seeing at the bottom of this slide right here. The nucleus, it is the largest, most obvious of all of the organelles if we're looking at these cells under the microscope. It is rather spherical in shape, and we can see that, so we're just looking at kind of a cross-section of this particular one. And we like to think of the nucleus as the control center of the cell, or the brain of the cell. And the reason for that is because most of the DNA, most of the genetic information is going to be inside the nucleus. And so it has the information, and it's going to be using that information to control or direct what the entire cell is doing. And that would be something that it does pretty much round the clock. If we look at some of the structures that we do have with the nucleus, we do have a lot of times this dark spot that shows up when we do stain the cells and we look at them under the microscope. This dark spot is what we call the nucleolus. The nucleolus is going to be where our RNA is synthesized. We have different types of RNA, but that's the primary location where we see it actually made. Other things about it, we do have the nucleoplasm. The nucleoplasm is the jelly-like substance that we would find inside the nucleus. And then the nucleus, besides just having a membrane around the outside, is actually a double membrane. So we do have two membranes. We call it the nuclear envelope, and this would surround the outside. So it's keeping that DNA inside, but then also the DNA has to be accessible because we're going to use it to run the cell. So there will be little pores, and we can see that mentioned here. These pores, these little holes, they're going to allow things to go in and things to go out. So if we think of the DNA as the blueprint for the cell, we're going to have molecules that have to go in, read the blueprint, and then take the information back out to the cytoplasm where the rest of the molecules are kind of ready and waiting on that information so that they can do something with it.